Non-diagnostic clinical assays performed during clinical trials provide investigators with critical information about underlying genomic and proteomic defects that may be driving the disease state or whether and how the drug is working in a patient's body. The assays can detect defective genes or proteins and determine whether the dosage is sufficient to induce a measured response that rises and falls over time, whether the drug is modulating the intended target, and what changes are seen at the subcellular and cellular levels. The quantified pharmacodynamic biomarkers are linked to the therapeutic efficacy to reach an understanding of the pharmacology of the new drug class. These assays must be conducted to rigorous standards to ensure that they can produce accurate, reliable results that correctly inform decisions by NCI programs and local investigators. Because non-diagnostic clinical assays are not regulated like diagnostic assays, many of them perform poorly in terms of accuracy and reliability. And sometimes, such poor performance is never corrected. The mission of the FNL's pharmacodynamics program is meeting the important significant challenge of accurately and reliably measuring molecular drug effects. The Scientific Standards Hub resource provides guidelines, best practices, and peer-reviewed examples for scientists working in drug development at mid to late preclinical and early clinical stages. We include SOPs to ensure rigorous and reproducible measurements from patient specimens using validated assays and procedures proven to preserve the analytes in those specimens. These assays are used to understand drug action, so good assays inform decision-making and bad assays could ruin a good drug. Reliable measurements depend on the rigorous development, optimization, validation, and clinical implementation of robust and quantitative assays. From specimen collection to data reporting, the use of validated methodologies and reference standards is essential to achieve valid assay results and to guide successful drug development. We encourage you to review these standards and implement them in your laboratory. Get in touch with us with any feedback or questions.